we were scheduled to have it a few weeks ago, but again, the class reps reached out to me saying you had a cut or something was going on, so you could not have it at that time. And that's why we're having it at quite early in the morning, just trying to make up. And the class is Yersinia, Francisella, and Pasturella. And I'm hoping that not just these lectures, you've not missed any other. If you have, make sure you're able to make it up with the lecturer, and if you're having any difficulties, please approach us in the department. So for those who have not yet met, my name is Marianne Morethi. I'm the chair, Department of Medical Microbiology and Immunology. So we could have met once or twice in the corridors. In case you have any issues in the department, you want clarification, you're looking for a mentor, or even looking for research opportunities, you can always reach out to us. We have a quite vibrant research community within the department and within the research institutes at CAVI and at UNITID, where we are heavily involved. And one of the aspects of our research is what you're seeing on the slide right now, and this is what we call zoonosis, and that's actually the backbone of our lecture today. We're looking at how our environment, animals around us, interact with us to cause some health challenges or difficulties. And this is what the aspect we're calling as One Health. This is actually the new kid in the block, the most latest type of research you can get involved in. And as you're seeing the current pandemic, coronaviruses, where we still have that aspect of, did it originate from a bat? How did it jump species? And this is all that encompasses what we mean as One Health. So how all these interact, and in particular interest, how they cause human disease. So, I don't know why my slides are not moving. So as I mentioned, as you're calling them zoonosis, some textbooks will call them arboviral infections. We're looking at viruses, and I'm sure you covered that with the virology group, and I'm sure they mentioned some of these things. And one of the topics I've mentioned is this emerging, the new infections that are coming up, and also re-emerging infections. Recently, we saw polio outbreak, I think, was it in Malawi? And that, you know, we had all thought we have gotten rid of polio. So that's something that we need to, sorry, please zoom into your slides. You cannot see, I'm already on slide mode. So it means something wrong with, sorry. Is there an issue with Zoom? Because I was on slide mode, but I'm seeing you cannot see.
It's okay now.
Yeah, so let me work with it on this way. So I've come out of slide mode. We're going to look at it this way. So yes, for those who do not see, our topic of the day from your timetables is Yersinia, Francisella, and Pasturella. And you're going to see how all these are linked or classified as somehow as zoonotic infections, because again, they have some animal origin or vector that needs, before it transmitted to humans, need to either be housed in a tick or in an animal, but we're going to look at that in more detail. And that's the aspect we're calling as One Health. And I'm sure you guys, as mentioning, you've covered these when you looked at virology, where you looked at emerging and re-emerging infections, now which is a major threat to global health. And apart from this, I'm sure you've been keeping up with the news. Actually, the most major concern at the moment is antimicrobial resistance. That's a topic for another day, but that's something we are quite concerned about, where we're developing resistance to all types of medication. Again, there are several factors because of that, and we need to keep an eye. So again, apart from the AMR, we, as a topic of the day, the zoonotic infections. And as I mentioned, they must have a vector or a vehicle that facilitates the transmission of these infections to humans. And it could be arthropods, could be birds, animals, or human to human. The spectrum is quite large. We're going to look at even ticks. Ticks have been known to transmit some type of infections. You're going to see when you're looking at Pasturella as well. And again, there are several factors that may be influencing these, and some of the factors could be environmental, ecological. We seem to be encroaching over and over again some habitats that that should not be encroached. For example, forested areas, or having a very intimate interaction with the animals. I'm sure when you travel to the villages, you'll find that in one hut or compound, animals of all sorts are in the same spectrum or the same structures where humans are sleeping. So there's quite a bit of interactions. And also now a major concern is also some of these emerging or re-emerging infections or pathogens can be harnessed if they go or fall into the wrong hands and used as bioweapons. And this is what, again, I'm still introducing my topic, but I'm still introducing my topic and just laying foundation that apart from looking at the pathogenesis of these infections before you look at them, we need to understand the seriousness of some of these organisms, such as yersinia, which is one of the first pathogens we're going to look at. And a major concern, apart from being emerging or re-emerging infection, a zoonotic infection, if it fell into the wrong hands, will lead to bioweapons. Again, what makes a bioweapon is a pathogen that could be quite infectious. We may not have developed any immunity towards it. So if it's introduced to a population, can cause quite severe trauma. So again, this is something we need to be quite aware of. And I saw even in the news last week, there are some labs in Russia, some labs also in Ukraine that house some of these dangerous pathogens, one of them being yersinia, another one being Ebola. And there's this constant fear that Putin may actually unleash some of these biological weapons to his enemies. So again, just showing the seriousness of some of these bioweapons. And some of them I've listed here. These are, as you can see, they're listed as category A, meaning they're the most dangerous to be in any public hands. So the labs that actually house some of these pathogens are quite well regulated. And as you can see, we have two that you're going to cover today. That is Yersinia pestis that has been known to cause, it used to cause the Black Death centuries ago. And there's some labs that still have this pathogen, and also Francisella that we're going also to look at, that is also categorized as category A. So if it fell in the wrong hands, can be used as bioweapons. And as I mentioned, there's a constant fear that this can be unleashed in Europe with the ongoing war. And I'm sure, again, you've covered some of the hemorrhagic fevers. You looked at smallpox, and you're looking at virology. And with my colleagues, if you've not covered yet, you're going to look at Clostridium and also anthrax, which again has been known to be a bioweapon. And the seriousness of this, I was just reading the news the other day. I was looking at the New Scientist magazine, and I noticed there's a lab actually in America that had some lapses in how they are storing these pathogens. Again, for researchers, we store some of these things because we want to understand them, maybe develop better vaccines, and also in case they fell into the wrong hands, we have vaccines that can be given to people, given to our soldiers, so that they are protected. So that's why some labs are commissioned to be able to house some of these pathogens. And these labs are normally categorized as category 3 or 4 biosafety labs. We have one at Unity, which is a category 3-4 that we are still working on. We still need to work out on the airflow of that lab, because we want to start working, looking at coronaviruses in more detail, HIV, TB, and all that can only be done in a biosafety lab of this level 3 and 4. But if we come to CAVI, where we're doing most of our research, we have category 2, where we're able, again, to look at some pathogens that are of less serious nature. So here in this magazine, you'll see that CDC actually shut one of the labs in America because of lapses of how they were storing their Yersinia pestis and their Francisella pathogens. So again, showing how seriously we need to take these pathogens that we're going to look at today. So that's just a preamble, but at the end of this lecture, again, it's a short lecture, so fear not. We're going to look at the general characteristics of some of these serious pathogens, known as we've introduced them as have a zoonotic origin, can be used as bioweapons if they fell into the wrong hands, and they need a vector, or in simple words, a vehicle, before they're transmitted into a human. So we're going to look at Yersinia, Francisella, and Pasteurella. I want us to look at their pathogenesis, how do we make a good diagnosis, treat, and also prevention. And as a researcher, I'm quite sad that we don't have a vaccine against any of the three, but as you can imagine, there's now a race to make better vaccines. Again, we need to have people protected in case, again, we have these pathogens reemerge in a bigger scale, or as I mentioned, the constant fear of them being used as bioweapons in a population. I remember I did some of my research in the United Kingdom when there was the Gulf War happening. I think it was Saddam Hussein and George Bush. And I remember in the place I was working in, my lab manager and everyone around the lab, and also some soldiers were given orders that they have to be vaccinated against smallpox, because there's that fear that Saddam Hussein may unleash these bioweapons to the soldiers that were fighting that war. And also the people where I was working in, medical doctors, who also needed to be vaccinated against smallpox in case they needed to treat some of the soldiers. So just really emphasizing the seriousness of not being able to handle some of these pathogens appropriately. So for me, always a solution is good if we can have a universal vaccine. And you've seen now we can develop vaccines in a much faster way. We have the mRNA technology now, where we can use that as a backbone to deliver different vaccines in a more appropriate way. At the moment, actually, I normally have a lot of stories. At CAVI, we are starting a clinical trial in collaboration with Moderna, and IAVI is standing for International AIDS Vaccine Initiative, where they have used the mRNA backbone that is being used by Moderna in the Moderna vaccine. And here we are incorporating some aspects of HIV and then using that to generate antibodies, broadly neutralizing antibodies. I don't know if you've done immunology yet. And again, then infuse this to the population we want to investigate. This can be used for HIV vaccine of the future. And we have very good hopes that actually we may be onto something. And I believe in the next few years, we'll also have a HIV vaccine with all these discoveries. So watch that space. So let's look at Yersinia. As I mentioned, it's a category A bioweapon, bacteria pathogen. And when you read Yersinia, this image always comes up, and I've seen it in movies, and it's actually shown in horror movies. But actually, this was the PPEs used in those centuries by medical doctors and people treating anyone with Yersinia. It's highly transmissible, and they needed to wear such attires so that, again, they don't breathe in the pathogen. And then, it is to cause mass deaths in the cities, particularly in Europe. So they needed something that will also prevent them from smelling the death smell, the rotten flesh. So that's why their PPEs were like this, but again, looks quite scary. But I guess when they make movies of coronaviruses and they show the PPEs we are today, the future generation may think that was quite a scary way to dress up. But anyway, Yersinia, it's an enterobactericide, which again, you've been covering with my colleagues. And there's several species under this family. There's Yersinia pestis, the most common, but there's also Yersinia pseudotuberculosis and Yersinia enterocolitica, which again form the species that are under Yersinia. So today we are just going to look at Yersinia pestis, but I believe, and I want to give you some homework. I don't know that you get homework, some reading materials that you can read more on Yersinia pseudotuberculosis and Yersinia enterocolitica. So as I mentioned, it's an ancient plague. We saw it in the early centuries would wipe out an entire population and also wipe out an entire generation. And as I mentioned, this was the PPEs when we were dealing with Yersinia. And again, I've mentioned why they had to dress up in such manners, and actually in the beaks that were using, you'd put some herbs so that they don't smell that rotten flesh within the population. And it has quite a history. If you're interested in history, you can read more the history of medicine or outbreaks. Here I've just summarized in the early centuries. If you watch movies if you like historical movies, I love historical movies, you'd see populations in Europe, in the UK, would actually literally be wiped out by this. And that's what they used to call the Black Death. We've not had major outbreaks of Yersinia, of the plague or the Black Death. And again, the jury is out there. What could have changed? Did we develop enough antibodies at that point? Did we evolve mechanisms to prevent us getting serious infections? But of recent note, this is a map from 2021. There's been a few outbreaks, and I remember sitting with Prof. Anzala and teams at CAVI just analyzing what's happening in Madagascar. There was an outbreak of this Black Death at some point, and also a few pockets have been reported within Africa, South America, within America itself, and also in Europe. So this is something we still need to watch out, understand this pathogen. If you can be able to get an appropriate vaccine, the better. And we can see what we need to understand, why is it re-emerging at this time and point where we've not had an outbreak in several years. And a recent example was in Madagascar. Again, I'll leave the jury out there. It's something we're still discussing. So how is it transmitted? Or how was it transmitted at that point? It needed vectors, and one of the major vectors here were rodents. And again, these rodents had fleas, and this is one of the fleas. And I'm sure next year you're going to look at parasites and entomology, where again you're going to look at Yersinia, but from the flea point of view, and you're looking at how these fleas carry the pathogen and then infect, or they're carried by these vectors known as, for example, the rodents. And you know how intimately rodents interact with humans and can then introduce this to a human population and cause this outbreak. So general characteristics, as I mentioned, it's a bacillus, Enterobacteriaceae, and it has a zoonotic nature, needs a rodent for it to be transmitted to humans. And again, some of these terminologies should not be new to you. And again, if I would ask you, what does it mean to be gram-negative or positive? I believe you can answer that. And if you've not had the good practicals that we normally give, let me know, or talk to the lab team to make sure that they're able to demonstrate to you the differences between gram-positive and gram-negative. For the topic of the day is a gram-negative bacilli, cocobacilli. And when we use the staining, Jensen staining, we'll see that it has a bipolar staining. So it stains at the tips of this bacilli, and this helps us identify it quite well in the lab, or where you're identifying it. And I'm sure at this point you're working in a category 3 or 4 biosafety laboratory, again, to reduce any infection to the lab person or the person investigating it. So in textbooks, you see it has that safety pin appearance that helps us identify it when we do a gram staining. It's also a facultative anaerobe, and I'm sure, again, this terminologies has been explained to you, and if not, we can discuss that when we're going through the discussions. So let's look at different virulence factors. Hello, class, have I lost you? Hello?
We can hear you.
Okay, I realized it was not sharing. I think I had reached here. Virulence factors, sorry about that. Virulence factors of the yersinia. I don't know what happened to my transmission. This is where we're looking at the pathogen in more detail. And we're looking at what virulence factors that make it be able to survive the host. And I'm sure, again, you've covered this terminology over and over again. In simple terminologies, a virulence factor is what makes it fit enough to survive against the host. Remember, the host will mount a very strong defense, immunological defense against any attack, any pathogen per se. So these pathogens have been able to evolve features that help them counteract the immune system, in other words. They have, first of all, and I'm sure you've seen in most bacteria, they have a capsule which is antiphagocytotic. So it prevents them, in simple words, being digested by eaters such as macrophages, innate immunology cells that are able to digest pathogens and then present them to T cells and B cells so that they can get rid of them from the body. So they've evolved to have capsules. They have different antigens. Again, these antigens are produced either to create some toxicity to the immune system, in other words, make an immune system paralyzed, cannot mount the proper defense. Again, these antigens also assist in preventing phagocytosis. It produces, as I mentioned, different toxins such as LPS, liposaccharide, which is an endotoxin. Again, makes it quite toxic for different layers of the immune system from effectively mounting any response. They also, virulent factors assist this pathogen survive. So it's able to extract nutrients that it needs. It's able to reproduce. So here it has the ability to absorb iron using different parameters within itself. It has fimbriae, which again assists it in attachment, and also other enzymes and other protein factors such as ribnolysin, which enables again to digest the different tissues so that it can move or get absorbed through the tissues of the human host or the host where it's infecting to be able to cause pathogenesis. So always look at virulent factors. This is an exam question we bring over and over again. An example is, like now I could ask you, name for me 5 virulent factors that, for example, mycobacteria may have, or in our example here, yersinia may have, or any other pathogen you've looked at. And I just want you to know virulence factors is what assist this pathogen evade the immune system, be able to extract nutrients, be able to reproduce within the host so that, again, it can survive. It's transmitted. Let's look at the transmission events of the pathogenesis. And remember, I mentioned that it needs a host, and it starts off with an infective flea. And again, you're going to see this cycle again when you're doing parasitology next year. But again, let's just look at that. It starts with a flea that carries this yersinia, causes no effect on the flea. And then because of close interaction, you can have wild rodents or even rodents that you find within the forest or within the normal environment can be infected with a flea that has yersinia. And remember, there's quite close interaction now with rodents. If, for example, in an hygienic area or places that has some quite forested areas. And these then wild rodents can directly transmit yersinia pestis to the human being, and again causes different pathogenesis. Again, that's one direct transmission. We can also have domesticated rodents, again, the ones you find in our compounds, carrying the flea and can have, again, direct contact with a human and cause also what we know as bubonic plague. This then infects the human host. And once this is the human host is infected, we see different pathogenesis. So we have this sylvatic cycle, again, from wild rodents either found in the wild or in forested areas. Then you have the urban cycle. So it just means domesticated rodents that you find all over cities or in different compounds that again can directly infect the human and cause an outbreak of yersinia, where we see quite big pathogenesis here, or symptoms, again, which we're going to look at in the next slide. And then we also have secondary, sorry, because I'm not on slideshow, I have to keep moving my slides so that you can see better. Sorry, this is a bit small, but again, now from human to human, again, it's quite transmissible, may progress from person to person, causing symptoms such as pneumonic plague. Again, highly transmissible, goes to the lungs as airborne droplets and causes, again, severe outbreak within that population. So you can see the different cycles where we have the sylvatic or wild cycle, the urban cycle. This is the primary host of patient X, who has quite different symptoms, but then may transmit to someone who's may not have been in contact with any rodent, but cause what we know as pneumonic plague. And this is what we see outbreaks within cities and in those early years in different centuries. So bubonic plague in humans, again, highly contagious and causes buboes. And these are like huge boils that we've not seen before, because we've not seen any of these outbreaks in several centuries again. Look quite black in nature and can cause quite internal hemorrhage and causing severe, very quick death in a population. Again, you can see the route of transmission. We have the close contact with the rodent, which then carries this flea, which can lead to this bite on the human, when the human comes into contact with that, and then causes this bubonic plague. But unfortunately, this human may transmit this through airborne droplets, causing pneumonic plague. Again, quite highly contagious. It primarily infects the lungs and again causes quite quick death. And from what we've seen from history, it usually has 100% mortality once you get this infection. And as I mentioned, some of the symptoms here, these are old images. I had to really look for them. Bubonic buboes that I've mentioned. You can see the hemorrhage that causes this darkening as well. This is septicemia, and of course, quite a quick death. And you can see why the medics at that point would dress up in such PPEs. You can imagine a whole town having this. It would take 3 to 4 days before we, from patient X to having almost an entire neighborhood wiped out by this. And that's why I think they called it the Black Plague or the Black Death. And at that point, they didn't have any interventions. So how do we make a lab diagnosis? Here again, if you're exposed, you can take a suggestive history where you've been, you've traveled to high forested areas. Again, always be aware these are highly infectious pathogen. So if you're collecting samples, we need to make sure that you're well dressed in appropriate PPEs to minimize the creation of aerosols. And the specimen you can take here are vast. You can take some blood, you can do some bubo aspirates from those buboes that I've mentioned, you can take some sputum if you suspect a pneumonic plague, and any other sample that, that can help you get that diagnosis right. And again here, these are common examination question. For example, here I'd ask you, walk me through the laboratory investigations of yersinia. So always start off with the specimen that you could collect. How do you collect it? And then how do you transport it in the lab? That's all the places we look for marks. So once it's in the lab, make sure, first of all, we normally do a macroscopic investigation. So what are you seeing with your naked eye? Make sure you note that in your lab book, If it's sputum, is it yellow in color? Is it bloody? If you're collecting blood, again, make sure the volumes are quite right. And for examination's sake, always mention this a macroscopic investigation before then we move to the microscopic investigation. So here's where you prepare your gram stain. And as I mentioned, the coccobacilli, paleomorphic in nature, paleo meaning it has quite interesting shape. So it's not your normal coccobacilli or circular in nature, as is, so it's quite elongated, and how it stains has a safety pin morphology, where once you stain, you see the different corners where the gram stain, where it pops up. So having that bipolar staining, or what we call safety pin staining. And I'm seeing a question here, what does pleomorphic means? And it's just a word that we use to describe cells that have different characteristics in shape, in size, or even color. Here's where I'm mentioning that safety pin appearance. So again, in your category 3-4 lab, you may want to culture, and here we would use a blood egg or McConkey, or you can also use specific yersinia selective medium. Again, making sure that you're well protected as you're working with this pathogen. And it takes between 24 to 48 hours before we see our colonies. And if you're using a blood agar, and I'm sure you've come across all these agar plates, and if you haven't, let me know so that I take through the lab team to make sure that they're able to demonstrate this to you. We would see small, shiny, non-hemolytic colonies, or if you're using McConkey, you may see translucent pink colonies. And these color changes are all influenced by the different ion that they're extracted from maybe the blood agar plates or the McConkey, or the different oxidizing effects as they extract nutrients from these agar plates. So lab, how do we confirm that? We normally use biochemical tests. And again, I will not go through all these biochemical tests because I believe you've come across them before. The catalase test, again, here we use the hydrogen peroxide. It's catalase positive. It, as you see, it's a facultative anaerobe, so more likely be an oxidase negative. And again, we can use different, apart from the biochemical tests, and these are the tests I myself prefer. Main choice here being the molecular tests, such as PCR. It's quite direct. Within 4 to 6 hours, you have your results, instead of the traditional cultures, which, as you've seen, can take 24 to 48 hours later. And this is actually the most, if your lab is equipped with all these reagents and the equipment, molecular tests will always be the way to go, but also there are rapids that have been developed, and also antibody tests that have also been developed to help us diagnose this yersinia. And you can see on our blood agar plate, our shiny non-hemolytic colonies, they're white in nature, is what you'd be able to observe if you're making the lab diagnosis. So here is what we see. Bipolar staining, as I mentioned, the safety pin nature, the dark staining at the very ends when you're using the right staining as well. And this is from a blood sample from someone that had plague in those early centuries. So how do we treat it? At that point, they had not discovered, Fleming had not discovered antibiotics. So I guess there's no survival rate there. You get it, it had 100% mortality. You could see the medics at that point to try and just make sure they're giving supportive care. But now we have enough antibiotics that can be able to treat any outbreaks of yersinia. But as I mentioned, we are on a ticking time bomb where we are dealing with AMR, that is antimicrobial resistance, and we're now starting to see resistance in our antibiotics. So again, really emphasizing we need to be able to do vaccine research and research to help us eliminate and eradicate some of these infections. So prevention, we need, of course, we start off our environment. How are we doing with the rats and fleas? We live in hygienic environments. And I believe we have really changed in terms of I'm thinking in the 10th, 14th century, they may have not discovered the sewage system of how to, make sure that cities, towns have an appropriate ways to collect their garbage and sewage systems as well. So maybe that's in these newer centuries or where we're living at the moment, we've been able to take control and in charge of how our waste products move from one place to a central collecting area. So that could be one reason we don't see these outbreaks as frequently as before. I mentioned also we may have evolved enough antibodies at that point to be able to control some of these infections. And yeah, the debate is on why we don't see major outbreaks anymore. If you ever see an outbreak, of course, we now know how to isolate and how to handle patients who are highly contagious. I think Corona has taught us one or two things, and have even better PPEs, not wearing those scary looking masks. And of course, for me, quite passionate about vaccine and vaccine discovery. We need to be able to develop an effective vaccine that can help us one day say we've eradicated plague or the infectious agent Yersinia pestis from the world. We've done that with smallpox, and I'm hoping that even with Yersinia, we'll be able to do that. And again, at this time and age of bioweapon and bioweapon discoveries, we need to revive this research. I'm seeing someone asking, what does plague mean? Plague is the disease that is caused by Yersinia pestis, can either be bubonic plague or pneumonic plague. And I think I've walked you through what the different pathogenesis or symptoms that you may see from this. Okay, as I mentioned, our homework or take-home homework is to look at Yersinia enterocolitica and also Yersinia pseudotuberculosis. I'll send you a few notes, and I would want you to read this in your groups or your quiet time, just to see other subspecies under the Yersinia family. So moving on quickly, let us look at Francisella, another zoonotic infection. And you can see here a rabbit. In some textbooks, they normally actually say Francisella causes rabbit fever. And you see someone here mowing their lawn. And why this is important, because some wild rabbits normally, and particularly in the West, they make holes. And for people who are working in this such an environment, may interact with those holes or come across these rabbits. And these rabbits may be carrying the Francisella pathogen, and that's how the transmission effects occur. And you can see, of course, the flea here and the ticks are responsible in carrying this Francisella pathogen or bacterium from this vector, this animal, to the human host. So seeing again why we are terming them as zoonotic infection. As I mentioned, I love the history of medicine and infections, and Yersinia was discovered in 1922 where an American bacteriologist, Edward Francis, I guess that's it's called Francisella, and the first recognized was Francisella tularensis, by then it was named as Bacterium tularensis, and is a causative agent of the infection caused by Francisella, and this infection is what we call as tularemia. And maybe you may not have heard this word before, but it's when you see it again, it's an infection caused by the Francisella pathogen or bacterium, and you've seen the route of transmission. Here's an interesting pathogen. I'm usually interested in the immune system and how it interacts with pathogens here. And here, these blue cells are macrophages. Remember, macrophages, if you've done immunology, are part of the innate immune system. And whenever there's an infection, they're mounted, they run to the pathogen itself. And what macrophages do, they're big eaters. So they try and engulf the pathogen. But Francisella is quite interesting in that, and I'm sure you saw that in also Mycobacterium, the macrophages are unable to digest them. And you saw that, as I mentioned, in Mycobacterium. So, for example, in Mycobacterium, the bacteria itself lives inside the macrophage and quite comfortably per se. And this is also what we see in Francisella. The Francisella has a capsule that is antiphagocytotic, and the macrophages are unable to digest and present this to the T cells and B cells. So that's something interesting for us to note. So classification, as I mentioned, we are looking at Francisella, and the subspecies here are quite many. So if you come across them in different textbooks when you're reading, they all belong to this family of Francisellaceae. And the most common is the Francisella tularensis that causes some skin infections that you're going to see in the next few slides. So what are our general characteristics? Again, here the common thing, we're looking at gram-negative coccobacilli. They are facultative anaerobes, intracellular pathogens. And why they're intracellular, you've seen that they can live inside a cell. And one of the cells I've mentioned are macrophages. They're non-motile, meaning they lack the flagella, so they need other mechanisms to help them move from one cell to another. And they can use the fibrinolysin, which helps digest different cells so that it can continue propagating as it causes the infection. They are aerobes, so they need oxygen to be able to breathe in. And this you can see again also they're non-spore forming. So epidemiology, this is an interesting map, and I thought I should show you from Nature Medicine or Nature Microbiology. And you can see the different outbreaks of Francisella tularensis. It's one of the few maps you see an infection that's not been seen in Africa. And I thought this is interesting to show you. I see that Francisella is mainly seen in the Western world, and both in America and in Europe, so tularensis, and the other subspecies such as Olactitica. You see it's Russia and different areas, and also some areas in Australia as well. So that's something interesting to note, the epidemiology of that. Okay. My slides are refusing to move, sorry. I'm seeing a question here, explain the facultative intracellular pathogen. I think I've mentioned that. They can survive within a host cell intracellular. And an example is the macrophages. Remember, these are immune cells that are mounted by the immune system once it's challenged by a pathogen, and they're able to live inside that pathogen, that cell, and if they are living within that cell, they're able to extract nutrients, they're able to reproduce within that cell, so that they can actually function within this. They need these cells to be able to function. Remember, they're non-motile, meaning they need mechanisms to move, and one mechanism is to live inside a host cell intracellularly, so that these cells can help their movement to cause the infection. So hopefully that's a better explanation. And another question here, where we don't see it in Africa. Good question. I'm not very sure why. One could be our environment, the climate as well. We may not be seeing these ticks or fleas that cause or transmit Francisella within sub-Saharan Africa, within actually the entire Africa. And maybe that's why we're yet to see any of these interactions or this infection of Francisella. So again, we can read more why we don't see it in Africa, but I believe that could be one of the major reasons. So again, we have the same item or pathogenesis of transmission, where we have either flies or ticks, and these ticks can are found in domesticated animals such as dogs and cats. And of course, these ticks can also be transmitted or infect both rabbits and rodents. And once this has occurred, because of direct interactions, particularly domesticated animals, if it's an infected flea, can also infect this human and causing this infection. We also have seen mosquitoes may directly bite a human being. This mosquito may be carrying Francisella as well and cause that infection. Again, these rabbits being an aerosol may create some aerosols, which again may be breathed in by the human, causing again the infection. So you can see this route, this zoonotic route of infection that needs interactions with either wild animals, rabbits, rodents, or even our domesticated animals, dogs and cats may be carrying the ticks that may cause that infection. And you can see when some mosquitoes may carry this. And these are, again, ticks and mosquitoes that may not be prone to our environment at the moment. So once this tick bites you or you breathed it in, we see different pathogenesis and how it spreads through and can cause septicemia. We see abscesses, particularly in the skin, may cause some digestive interactions as well, causing diarrhea, and may also lead to some, where the bite was, again, some darkening of the skin, and also you see conjunctival interactions. And when you be looking at dermatological infections, tularemia may be one of the infections that you may see. And you can see here it causes a skin ulcer. I believe maybe this is where the tick transmitted either by the dog or the cat may have bitten this human host because this type of interaction. So how do we make a lab diagnosis? Again, we always start, what specimen are you interested in? It could be secretion, you could do a skin scraping, you could also maybe even do an aspirate to take the blood or sputum, depending on how your patient is showing symptoms. Do a simple gram stain, and from here, we'll be able to see that it's gram negative, as we saw, Coccobacilli, as you can see here. And from the gram staining, we'd want to confirm what we're seeing. So that's why we do the cultures, and we can use either the cystine heart blood agar, and here we can use charcoal yeast agar, which prevents other bacteria from growing around, so that we can have pathogen of interest growing between 24 to 48 hours later. And with that, we may be able to confirm that indeed what we're seeing is Francisella. It's always important to hold it for around 10 days because it's quite contagious, to make sure that it dries off and dies off before we discard it. And we always bleach the area around where we've worked, make sure we are cleaning up properly, handling these infectious agents in a proper laboratory to prevent misuse. So what do we observe following our culture? If we use a CBA agar plate, that's a chocolate blood agar plate, we may see tiny gray-white colonies, as you can see here. If we use a cystine heart agar, these are all commercially available agars, we may see a greenish blue colony. So this is something you need to have in mind if you're making a diagnosis as a microbiologist. But of course, if you can take a step further, either to do some histochemical staining, and of course the lab diagnosis of choice PCR, which makes you make a direct confirmation and at a very quick state. And here is just a paper I was reading where they used PCR, again, state-of-the-art PCR, to be able to diagnose what type of Francisella they got from their specimen or their sample. This was an outbreak again in the United States, and they published and were able to show using PCR, they were able to diagnose that the patient had Francisella tularensis, and again, even move further to look at the different subspecies. That's why I am very pleased when I see results presented from PCR test. But again, in our nature, our clinics here, we may not have some of these techniques and capabilities. So that's why, again, when we are teaching, we'll always make sure we take you through the lab staining, gram staining, which is the basis of microbiological investigations before we do our cultures, to be able to confirm that indeed your patient has, in this example, Francisella. Treatment, of course, we have different antibiotics that have been used in the West and have been quite successful. We don't have a vaccine yet. I know there's active vaccine research, again, because this can be weaponized and used as a bioweapon. So again, and why it's being used as a bioweapon, as I mentioned, it's highly transmissible and can cause severe infections in the population that is exposed to it. So last but not least, let us look at Pasteurella. And here we have cats and dogs. You may be exposed to this infection. Some call it the cat fever. I normally have 1 cat and 4 dogs. So these are things that I always have at the back of my mind. And I make sure they're quite, they're clean and don't have ticks or fleas. Pasteurella This is the family of this type of infections or this pathogen, and has different subspecies as is shown here, all the way Pasturella avium, canis, Lagmatis, Pasturella moticida, as well as Pasturella sensu. Again, all these are seen in domesticated animals, particularly, the canines, the dogs, or even the cats. So a general overview of Pasturella. They're again Coccobacilli. They are gram-negative and facultative anaerobes again, and therefore once you get infected or the tick bites you, it can cause where the bite has been, you can develop a wound. So that's something to look out for. And the major one that we see in our domesticated cats or dogs is Pasturella moticocida. It's most common human pathogen. And as I mentioned, the domesticated pets serve as major reservoirs. And and again, when it's found in the pets, in our pets, it's like a commensal, part of its microbiota or commensal, and mainly found in the upper respiratory tract of these animals. So either you get bitten or in close contact, in close contact with a human being, and develop a bite or a scratch, or even share food with these animals. Again, being a commensal, you could inhale or be exposed to the Pasturella pathogens. That's it's always important if you're going to go have domesticated animals, maybe avoid sharing foods. I've seen that in the West, where they treat these animals as their babies. And that's where we see some of these infections, mainly in the Western world, rarely in where we are at the moment. But again, as microbiologists, we be aware of these infections because again, you may never know. You may be posted to work in a lab in the US where maybe there's a Francisella outbreak or a Pasturella outbreak, and it's good to be able to know these pathogens. So a lab diagnosis, you could take a swab, you could take a sputum, blood sample. It's quite vast what you can use as a specimen. Once you have it in the lab, make sure you do your macroscopic investigation. Note that down. What do you see with your naked eye? Is any pus cells? Is it what's the color? What's the volume? All this helps us in making a proper diagnosis. And then when you do your gram stain, as I mentioned, they're gram-negative Coccobacilli. And when you culture, and you would culture the blood agar plate, they produce grayish colonies, grayish shiny colonies, as you can see in this blood agar plate after 24 hours of culture. Again, you may also want to do PCR in a lab that has this technology. Treatment, we have antibiotics that help treat Pasturella outbreak, both in humans and also in your domesticated pets, but always prevention and control, if possible, avoid very close contact with these pets, because again, as I mentioned, it's part of their commensals, part of their microbiome. So if they have this, it's easily transmissible to humans and may cause this either as a wound or, as we saw, the infection in humans. So just a summary, we've looked at yersinia, we've looked at Francisella, and the most common one is Francisella tularensis. We've looked at the different characteristics, gram-negative, they are bacilli, like Francisella it doesn't have the flagella, it's non-motile. It's an aerobe again, and you can see that it's the different biochemical tests, the different virulent factors. As I mentioned, we have the capsule, we have the pili, which, if you remember the beginning of your lectures, pili help attach to different cells. And remember, it needs to attach to these cells so that it can be able to move from one cell to another. So it has evolved all these mechanisms. And I mentioned found mainly in the US and comes about in the summer months. Again, people who study the environment, ecologists may give us better answers. And of course, we've seen rodents are a good reservoir, or even rabbits, once you're exposed to that. And in rabbits, you've seen that any rabbit infected by Francisella cause normally it's a capsid infection. So in humans, it causes a disease known as tularemia. Some texts call it rabbit fever. This what we see: fevers, headaches, ulcerative lesions that may be at the site of infection, and that can cause other wound type pathogenesis. And you've seen transmission here through the bite of the tick, or direct contact, or it's also airborne. Diagnosis, we've looked at culture, we've looked at PCRs as well, and treatment at the moment. Aminoglycosides have been shown to be quite successful. We've looked at Pasteurella, and the most common human one is Pasteurella multicida. Again, we've seen it's a gram-negative bacilli. It's non-motile, meaning it lacks the flagella and its other mechanism to move, has different virulence factors, just such as a capsule. Mainly it's been reported worldwide, but again, majorly in the Western world. And as we mentioned, it's a normal oral flora of both cats and dogs, which we have them as our domesticated animals. So we really need to be careful how we interact with them. And transmission, as we've seen in our lecture, either through a bite, a scratch, or if you have close contact, could be airborne, and you could breathe it in. And the different type of symptoms here, we see some skin changes, cellulitis. Again, you can see some necrotizing or abscess forming, which then can lead to secondary infections if it's not treated well. We've seen in the lab, gram-negative, we can use blood agar. Again, it's the agar plate of choice, and treatment here. Again, some treatment options that have been shown to be quite successful. So with that, I think at the end of my slides, I'll send you a few notes, particularly with our take-home notes. Maybe when we next meet, we can talk about why Francisella is mainly seen in the US and in the UK, not in Africa. So that's something to look about. And also want you to read more on vaccine research. Where are we in development of vaccines? Do we have any vaccines for parasites per se? I know you're going to do parasitology next year, but these are things we as also researchers, we're always challenging ourselves. Why don't we have vaccines even for fungi infections? We're going to look at mycology in a few weeks' time, but I'm yet to see a vaccine against, Candida, which can cause quite also major discomfort in humans. So I believe vaccines are a way to go, particularly when you're looking at antimicrobial resistance. If we can prevent the infection, the better. So allow me to end there. Any questions are welcomed. You can send me questions also on my email. You can contact me also on my Twitter page. You can find me at Doc Moreidi. I'm always tweeting some science thing or microbiology thing, and we can interact better that way. So with that, I know you may have a class now. I'm sharing my email. So thank you. And sorry for the background noises.
Thank you.
Yes, I'm seeing I should share slides. I'll do that. As a question, why isn't Africa affected by Francisella? That's our take-home homework. Okay, so then have a good day, class, and see you soon.